So 2 summers ago, I planted a tomato garden in my backyard. And I did this for 2 main reasons. First off, I really like tomatoes. And second off, I wanted to experience what it's like to grow something. And it was hard work. Because it was a particularly hot summer, my plants really suffered from the heat. So every day I would have to go out and water them. And my plants weren't alone. Tomatoes are the 4th most cultivated crop around the world. And the 2 most cultivated varieties of tomato are particularly weak to droughts. And worse, droughts are becoming increasingly more common as a result of global warming. Scientists aren't quite sure why tomatoes are so weak to droughts, but they have a trick up their sleeve that will help them figure it out. That trick is CRISPR-Cas9. CRISPR-Cas9 is a gene editing tool that allows scientists to make edits and cuts in genetic material. Cas9 is an enzyme that acts as a pair of molecular scissors to make cuts in DNA. One type of experiment that can be performed with Cas9 is a gene knockout experiment, where Cas9 is used to make a cut in the DNA to deactivate a gene or knock it out. For example, if we have a gene in a mouse whose function we want to figure out, we can knock it out, and if it, say, starts shedding fur, we know that the gene used to cause the mouse to keep its fur off. So how can we apply this to tomatoes? Remember, we want to figure out what gene in tomatoes results in drought resistance, and how we might be able to improve it. So what we can do is, using some tools, perform a gene knockout experiment on a gene we think might have drought resistance, called the SLNPR1 gene, to see if it really does what we think it does. There are 3 important parts to a gene knockout experiment. Cas9, which we've already discussed, the enzyme that will act as molecular scissors and cut the DNA. The next important part is guide RNA. Guide RNA is crucial to the Cas9 mechanism because it's like the hand that tells the scissors where to go, where to cut. Without guide RNA, Cas9 might make cuts in the wrong place, which could be disastrous for the organism. Cas9 encodes the location of 20 base pairs adjacent to the cutting site, which Cas9 will use to check if it's in the right place before it makes any cuts. Cas9 and the guide RNA together form a complex that can be used to edit DNA in organisms, but we're not quite done yet. Because DNA is so important to the organism, it's guarded within a structure in the cell called the nucleus. The nucleus is like a vault for the DNA, which is like the gold. Cas9 is like a thief who wants to sneak into the vault and take the gold. So the defenses of the nucleus are perfectly designed to keep Cas9 out. But we can give Cas9 an advantage by giving it a special promoter called the nuclear localization sequence. Even though the nucleus is a vault, it still needs to let things in and out, and what goes in and out is controlled by the nuclear membrane. The nuclear membrane tags proteins with a nuclear localization sequence, which is sort of like a secret password to the vault. If the protein has a secret password, it's allowed in, but without it, it's kept out. This keeps foreign material, such as Cas9, from entering the nucleus and messing with the DNA. But the nucleus didn't count on scientists figuring out the nuclear localization sequence. By giving Cas9 the tomato-specific nuclear localization sequence, it can get into the vault very easily. So with all 3 of our parts ready, Cas9, guide RNA, and the nuclear localization sequence, we're ready to insert these parts into the organism and see if our hypothesis was correct. And it turns out that it was. The SLNPR1 gene does in fact induce drought response in tomatoes. After being edited, the plants were subjected to drought-like conditions, such as high heat and loss of water. And the edited plants fared much worse than the unedited ones. The edited plants experienced broken stems, wilted leaves, and water leakage, whereas the unedited plants survived the drought with no problems. This is the first step to engineering a more drought-resistant tomato. With this knowledge, we can now overexpress the SLNPR1 gene, or even engineer in a whole new drought resistance mechanism. This is the future of agriculture, using tools like CRISPR-Cas9 to learn more about organisms, and then using this information to grow them in a sustainable and optimal way. So the next time I grow a tomato garden, my plants will be able to beat the heat, and so will all the other tomato crops around the world, all thanks to gene editing. Thank you.